Good evening, everybody, and welcome to our FB Live. This is Dr. Mamta Dike. I'm the director of Zenith Advanced Fertility Center. We are based in Pune at Wakat as well as at Koregaon Park. Um, Unfortunately, Dr. Pallavi is not here. We are having this series on male infertility, and we have discussed various topics on male infertility since we started this series, which was in the form of a question answer session. But since she's not here today, I am going to be taking the session myself, and let me see what I can get across to you today. So, if you all have gone through the FB Live based on this series, I think you'll know that we have covered a lot of questions, issues related to male infertility. Today, I am going to talk on just two very specific topics. These are two things which are hardly ever discussed, and I think that is why it is very important for you to get an understanding of what I'm talking about. It might be a little bit of medical jargon, but I think it is necessary for this to go out to the mainstream so that people can understand what is happening. Now, the two things that I'm going to talk about, one is about semen freezing, and the second thing is about the genetics of male infertility. So, first, let me touch upon semen freezing. Now, what is semen freezing or cryopreservation, and when is it required? So, when we collect a semen sample and we store this semen sample, by a specific method, there is a specific method by which sperms are frozen and then they are stored in liquid nitrogen. So, when we collect a semen sample and then freeze it and store it in liquid nitrogen with the intention of using it at a later date, that is called semen freezing. Now, when can you freeze the semen sample? What are the indications of freezing a semen sample? So, the first and the most common indication is that for whatever reasons, if while doing an IUI or while doing an IVF, if the male partner or the husband is not going to be available on that particular day, then the semen samples, which are normal semen samples of such husbands, are stored prior with the intention of using it on the day of the IUI or on the day of the IVF. That is the most common reason why a semen sample is preserved. Another reason is that when we go through an IVF procedure and the male, the husband has a completely normal semen count, many times we have observed that exactly on the day of the IVF procedure, it becomes very difficult for whatsoever reasons. There could be stress, apprehension, anxiety. For whatever reasons, the husband cannot produce a semen sample. And if we have withdrawn or drawn or retrieved the oocytes already on that day, the oocytes need to be fertilized with the semen sample on that day itself, failing which these oocytes will have to be frozen. And then we will have to again thaw these or warm these oocytes and then fertilize it at a later point when the semen sample is available. So, in order to avoid such a situation, we freeze the semen samples of the male partner so that just in case there is a problem in giving the semen sample, particularly on the day of the IVF, then we can utilize the stored semen sample that is already stored in the laboratory for this purpose. The third reason is that sometimes we have seen men who go into deteriorating levels of their semen or the sperm counts. So they start with a relatively good count, albeit a little lower compared to what the normal values are. But over a period of time, the semen or the sperms in the semen sample show a trend towards lower and lower counts. Now, this usually happens if there is some problem at the testicular level where the production of the sperms is getting hampered. And hence, before such a man kind of reaches a position where the sperms are really very less or absent completely, we tend to store multiple semen samples in such a case. For example, let's say that there is a oligoasthenoteratozoospermia, that is low count, motility, as well as some abnormalities, but there are sperms in the semen sample which can be used. And we have seen that the previous samples were slightly good, better counts, then and now slowly the counts have started decreasing, like let's say from maybe 6 million it's gone to 3 million, and now it's kind of on the lower side, trending towards decreasing. Then in such cases we would store multiple semen samples of the male partner, especially if they are planning to not do their IVF immediately, so that even at a later point in time, if there is real deterioration in the testicular function and absence of sperms, we at least have the sperms preserved. The fourth reason is if for some reason there is a detection of a malignancy, and the male needs to go through, let's say, a surgery or chemotherapy or radiation therapy, then under such cases, again, the semen samples, multiple semen samples are frozen. And of course, post pubertal males, even younger males, can thus freeze their semen sample. This is known as fertility preservation. With the increasing, promising cure rate in cancers, and the longevity increasing, mortality decreasing, many times after the initial phase has passed, all individuals start thinking about their fertility. And if you have the semen samples frozen, then definitely you can use this, because once there is exposure to radiation or chemotherapy, it will affect the gonads, and therefore it might affect the semen count, especially the semen quality, the sperm quality. And hence, prior to something like this, the semen samples are frozen. Lastly is, if you have very low semen counts, or if you have azoospermia, that is, azoospermia is absence of sperms in the semen sample. We've already discussed about azoospermia being of two types, either an obstructive azoospermia or a non-obstructive azoospermia. So if you have azoospermia and you have retrieved sperms from the testis, we advocate that this procedure should be done around the time of IVF 1. If not, at least it should be done in a laboratory where the sperms, if they are obtained through a microsurgical procedure, should at least have facilities for freezing, however less the sperms are. So if you have done a surgical extraction of sperms and there are excess sperms, then again, these sperms are to be frozen. So these are more or less some of the indications under which we ask the males to freeze their semen sample. Now, we need to remember that if we want to use the semen sample, like frozen semen sample, and of course, if you are using donor sperm samples, so if for some reason there are absolutely no sperms and the chances of finding sperms from the testis in the husband is very minimal, some couples have to resort to using donor sperms. Under such cases, obviously, the donor semen is always frozen, and there is an infectious disease screen that is always performed prior to freezing and after freezing, before the sperm can be used. And this is again one more indication where sperm freezing is advocated. So just to give you an overview, these are the conditions under which the sperms or the semen can be frozen and kept in the lab. The sperm should be frozen in an IVF lab, where there are all facilities for freezing them properly, for storing them properly, for thawing them properly, and making sure that they're processed in the correct way so that they can be utilized in a good manner for the fertilization procedure. Now, if you want to do an IUI using a frozen semen sample, then the count and motility in that sample should be good, because whether we like it or not, the freezing process is going to have some impact on the sperms. You're not going to get 100% retrieval of whatever sperms are were there initially before the freezing procedure. So you have to kind of filter it, what is going to be a little bit of damage or deterioration in the semen sample, and make sure that at least the total motile sperms that you have available in the final washed semen sample, which is going to be used for the procedure of IUI or IVF, should be good. So, for an IUI procedure, we definitely want at least a minimum of 8 to 10 million sperms, motile sperms, post wash. Though it has been said that even up to 2 million of sperms in the post wash sample can be utilized for IUI. We have many studies which has shown that the good rate of IUI success is available when you have at least about 8 to 10 million sperms in the post-wash semen sample. So this is what should be there, or the limit that should be there for your IUI sperms, a semen sample. For IVF, you need at least about 1 to 2 million. And for ICSI, even very few sperms that you have, especially if you have sperms that are taken from the testis, because ICSI is a procedure where you will take a single normal sperm and you will inject it inside the egg of the woman, which has been retrieved by the IVF procedure, by the procedure of oocyte retrieval. So in ICSI, even very few sperms, even a handful of sperms are good enough as long as they are live and they are normal, and you can utilize these sperms for the fertilization with the ICSI procedure. So this is a little bit about semen freezing. I'm not going to go into too many other details of semen freezing, but just to give you a gist about what semen freezing is all about. I'm going to go into a more kind of gritty subject, which is the genetics of male infertility. Um, I want to touch upon this is because genetics as a field is coming up in a big way, and there were many reasons which we would term as unexplained infertility, unexplained male infertility, unexplained female infertility, and we are getting some insights into the fact that there could be major or minor genetic issues, chromosomal issues, genetic issues, which could be a cause of this infertility. And that is the reason why we need to understand. Now, why are we really looking at this Y chromosome so much? Is it just an academic process that, oh, I should know everything about the genes, or will it have a clinical impact? Will it have an impact on the progeny? And what is when should it be done, and which are the tests that should be done? Now, what has been seen is that, so let's look at the issues that come up in the semen sample of the male, and let's look at what could be the genetic aberrations in such cases. So, let's start with a semen sample where you have a low count, low motility probably, but mainly an oligoastheno or less number of sperms in the semen sample. Now, again, oligospermia or oligoasthenozoospermia is can be a borderline oligo, that means slightly less number of sperms in the semen, or a severe oligozoospermia where the numbers are very, very less but they are present in the semen sample. Now, if it is a case of a severe oligozoospermia where the counts of the sperm in the semen sample only range between, let's say, anywhere between 3 to 1 million or even less than that, there is there have been found this has been found to be associated with some chromosomal issues in the male partner. Now, what is this? So let me see if I can use something. Yeah. All right. So what is happening is that, let's say I'm only looking at the sex chromosomes. So let's say these are two X chromosomes. So this is the female karyotype, and this is the male karyotype. This is the X and this is the Y. Now, sometimes, and of course there are autosomal chromosomes. Autosomal is the, not the sex determining, the other characteristic autosomal or somatic cell determining chromosomes that are present. Now what has been found is that there could be minor inversions, or there could be changes in the banding pattern of the chromosomes in a male who has either no sperms or who has very, very less sperms in the semen sample. So if you have certain areas where we call it like a, a change in the banding pattern could be present in any of the somatic chromosomes, there could be inversion in the sense, depending on how the chromosomes are supposed to be, you can get it rotated or inverted. So if such things are present in the male chromosomes, you may tend to have a low count or no sperms at all. Secondly, in the Y chromosome, there are certain areas which are coding for the presence of sperms, because ultimately it is the Y which is, important, and which gives rise to the presence of the male gonads and the production of sperms, etc. So if there are certain deletions on the Y chromosomes, the production of sperms is either going to get hampered or there won't be any production at all. So that is why it might be necessary to test men who have a very low count or no sperms in the semen sample, to check their chromosomes and to check for a test called Y chromosome microdeletion. Now, we always think that, um, the Y only has got to do with the expression of the gonads or the sperms, etc. But research has found that if there is a Y chromosome microdeletion, there could be a slightly higher impact in the neurological development and a slightly higher risk of cardiac problems in such males. So it's not just about getting your fertility straight. It is just about also understanding how the whole thing, because there are many people who come to us and say, can you tell us a reason why this is happening? And many a times we say it is unexplained, but it remains unexplained till the progress and the march of science gives you more and more reasons after delving into what the causes could be. So when we may not have had these reasons and causes some years back, but today we are understanding that there are certain patterns in the chromosomes of the male partner that can lead to problems of having a low count of the sperms. Not only will it affect fertility, but there's a small possibility that this could also affect health. And hence, it is necessary under certain conditions, which will be told to you by your fertility consultants, when it should be done and when there is no need to go in for it. The next thing is about azoospermia. Now, there are two types of azoospermias. Like we said, azoospermia, if you define it, it is absence of sperms in the semen sample. We have already discussed that there are two types of azoospermia. One is an obstructive azoospermia. So what happens in an obstructive azoospermia is that if this is the testis and this is the tubes, which you call it vas deferens. So there is production of sperms in the testis, but due to some blockage at some point, these sperms cannot come out into the semen sample. Now, if there is such a condition where The production of sperms in the testis is normal, but there is no presence of sperms in the semen sample. We have to do a scrotal Doppler or a sonography of the testis, the whole scrotum, and ensure the presence of the ducts called the vas deferens, which are connected from the testis coming out into the through the urethra, I mean by the side of the urethra, and which kind of help the sperms to traverse and come into the semen sample. Now, at times, these tubes, ducts, or the vas deferens are congenitally absent. So they've never been formed at all. Now, if there is a case where there is bilateral congenital absence of the vas deferens, that is on both sides, because we have two testes, one on either side. So, if the ducts from either side or both sides are completely absent, and this can happen only since birth, so it is called as congenital bilateral absence of the vas deferens, commonly termed as CBABD. If that is the case, there is a high chance that this male carries a gene of cystic fibrosis. So cystic fibrosis is a condition which can lead to a accumulation of a lot of thick mucus in many of the organs of the body, and can cause a lot of morbidity, and depending on the severity can really affect health and even life for that matter. So again, the cystic fibrosis gene is, like we say, we need two genes. We are a product of our genes coming from the father as well as from the mother. So if you have two genes, like the genes are present on the chromosomes, so if you have two genes, then there will be the person will be affected with the disease. If you have only one gene, which is a recessive gene, then you are called as a carrier. So if you are a carrier of this gene, there's a high chance that the vas deferens is absent since birth. There will be no disease of the cystic fibrosis because you're only a carrier. But if this male is married to a female who is also a carrier of the cystic fibrosis gene, then the baby can get one gene from the father and one gene from the mother, and the baby could get affected with the disease. So it is not just to understand whether the sperms will be, so sorry, my nose is scratching. So it is not just the question of getting sperms, and that is where I'm trying to get at is we are not here to just get sperms, get eggs, and by hook or crook just try to make an embryo so that it implants. That is not the basis, not the reason why IVF should ever be done. The reason should always be to make sure, to whatever extent possible, nobody can control it completely at all, irrespective of what tests you do, but to whatever extent possible, ensure that we are doing as much as we can to make sure that you know there is no passage of any possible issues or disease to the offspring. So, under such cases, again, so if there is a congenital bilateral absence of vas deferens, the male should get tested for whether they have the gene of cystic fibrosis. If the male shows the gene of cystic fibrosis, the wife should also get tested. So, again, a very important thing. It was said that the incidence of cystic fibrosis is very low in Indians, so it's not really a disease that is commonly seen here. It affects Caucasians to a much larger extent, and some Jews to a much Ashkenazi Jews and some other ethnicities. But research, especially from our own country, which is really doing absolutely great research at ICMR and the other labs in Mumbai, have been seeing that there are many novel mutations which are present. So a very important thing of what needs to be done, again, like I said, there could be some chromosomal abnormalities, there could be some major chromosomal abnormalities and in the male. And if you look at this, then you will be able to look at the embryos. We have the pre-implantation genetic screening available now. Of course, not something that needs to be utilized by all couples, but a very good tool, especially for certain cases where you need to check whether there is the normal set of chromosomes in the embryo that is present. So, like I said, there are certain cases, and I'm not going to go into the other factors, like there are changes in the heterochromatin areas. So that gets into too much of nitty gritty. What I just want to bring out in this talk today is that please talk to your consultant. Please try and understand whether doing a chromosome and a genetic test in the male, or for that matter under certain conditions in the female, for a different set of reasons, for the male for a different set of reasons, or for the couple for a different set of reasons, are required. But since we are talking about male infertility over here, that's the reason I'm talking about everything regarding the males. But if there is a low count or absence of sperms in the semen sample, please talk to your fertility consultant regarding whether any kind of a chromosome or genetic test is required to be done before you plan the treatment, be it IUI. But usually, with the kind of counts that I'm talking about, these males would usually require an IVF ICSI procedure. But in any case, depending on what the sample is showing, please talk to the fertility consultant whether a chromosome or a genetic workup needs to be done in the male in order to ensure that there is no problem in the chromosomes, and there is nothing that is likely to be passed down to the offspring, which might affect the health of the offspring in the future. So now this is a little bit about semen freezing and the genetics of male infertility. Genetics is a very, very deep subject, and we also always rush to geneticists to get an understanding. I think I'll call it a day, because I'm sure I hope I have not burdened you with too much of medical jargon, but there is really no other way, especially on an FB Live, to kind of explain these things. But even just a little bit of stimulation to understand that, please do talk to your fertility consultants and ask them whether any kind of genetic workup or chromosomal workup is required. So thank you for joining us, and wishing you all a great weekend ahead. Thank you. Bye bye.